Cell cycle and cell division. Introduction. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. Growth and reproduction are characteristics of cells, indeed of all living organisms. Such cycles of growth and divisions allow the single cell to form a structure consisting of millions of cells. Cell cycle. It is a sequence of events by which a cell duplicates its genome, synthesis the other constituents of the cell, and eventually divides into two daughter cells. Cell growth results in disturbing the ratio between the nucleus and cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleo-cytoplasmic ratio. Cell growth in terms of cytoplasmic increase is a continuous process. Duration of a cell cycle can vary from organism to organism and also from cell type to cell type. Example, yeast cell cycle duration is 90 minutes. Human cell cycle duration 24 hours, but cell division proper lasts for only about an hour. Phases of cell cycle. Interphase, called resting phase. Cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. It lasts more than 95% of the duration of cell cycle. M phase, actual cell division phase. Starts with nuclear division, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. Interphase, divided further into three phases. G1 phases, gap 1, S phase, synthesis, G2 phase, gap 2. Some cells do not divide further, exit G1 phase to enter an inactive stage called quiescent stage, G0 of the cell cycle. Cells in this stage remains metabolically active but no longer proliferate unless called on to do so, depending on the requirements of the organisms. Example, heart cells. G1 phase, interval between the mitosis and initiation of DNA replication. Cell is metabolically active and continuously grows but does not replicate its DNA. Most of the cell organelles duplicate. S phase. This phase marks the phase of DNA replication and chromosome duplication. Amount of DNA per cell doubles but there is no increase in the initial chromosome number. In animal cells, embryo duplicates in cytoplasm, DNA replication begins in nucleus. In G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. In animal cells, mitotic division is only seen in diploid somatic cells. Plant cells show mitotic division in both haploid and diploid cells. M phase, mitosis. Most dramatic period of cell cycle. Chromosome number in parent and progeny cells is the same, hence called equational division. Chromosome number in parents and progeny is the same. It is divided into two, karyokinesis and cytokinesis. Prophase. First phase follows the S and G2 phase of interphase, marked by the initiation of condensation and chromosomal material which becomes untangled. Centrosomes start to move towards opposite poles. The completion of prophase can be marked by chromosomes condensed to form compact mitotic chromosome. Each centrosome reaches at poles and radiates out microtubules called asters. The two asters together with spindle fibers form mitotic apparatus. At the end of prophase, cells do not show Golgi complex, endoplasmic reticulum, nucleolus, and nuclear envelope. Metaphase. The complete disintegration of the nuclear envelope marks the start of metaphase. Condensation of chromosome is completed and can be observed clearly under microscope, and morphology of chromosomes is most easily studied. Each chromosome has two sister chromatids which are held together by centromere. All the chromosomes coming to lie at equator. Each chromosome connected by its kinetochore to spindle fiber from one pole and sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as metaphase plate. Anaphase. Centromere of each chromosome split simultaneously and chromatids separate. Two daughter chromatids, now referred to as daughter chromosome, begin their migration towards the two opposite poles. The centromere of each chromosome remains directed towards the pole and arms trailing behind, leading to different shapes of chromosomes. Telophase. Final stage of karyokinesis. Chromosomes that have reached their respective poles decondense and lose their individuality. Nuclear envelope develops around the chromosomes cluster at each pole, forming two daughter nuclei. nucleolus, Golgi complex, and endoplasmic reticulum reform. Cytokinesis. It is division of cytoplasm at the end of which cell division gets completed. In animal cells, it is achieved by the appearance of a furrow in the plasma membrane. In plant cells, cytokinesis is achieved by wall formation, which starts in the center of the cell and grow outwards to meet the existing lateral wall. The formation of new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of two adjacent cells. When karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium, example liquid endosperm of coconut. Kinetochore is disc-shaped structure at the surface of centromere, serve as the site of attachment for spindle fibers. Significance of mitosis. Mitosis usually results in identical genetic complement in daughter cells. Growth of multicellular organisms. Cell repair. Mitotic division in apical and lateral meristem result in continuous growth of plants throughout their life. In some lower plants and some social insects, haploid cells also divide by mitosis. Meiosis. Specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. Meiosis ensures the production of haploid phase in the early life cycle of sexually reproducing organisms, whereas fertilization restores their diploid phase. Meiosis involves two sequential cycles of nuclear division, that is meiosis 1 and meiosis 2, but only single cycle of DNA replication. Meiosis 1 initiated after parental chromosomes have replicated. 4 haploid cells are formed at the end of meiosis 2. Meiosis 1 reduction phase. Prophase 1 typically longer and more complex when compared to prophase of mitosis. Leptotein. Chromosomes become gradually visible under the light microscope. The compaction of chromosomes continues throughout the leptotein. Zygotein. Homologous chromosomes called synapses. Synapsis is accompanied by formation of complex structure called synaptomal complex. The complex formed by a pair of synapsed homologous chromosomes is called bivalent or tetrad. Leptotein and zygotein are relatively short-lived compared to the packetine. Packetine. 4 chromatids of each bivalent becomes distinct and clearly appears as tetrads. Appearance of recombination nodule. The site at which crossing over occurs between the non-sister chromatids of the homologous chromosomes. Crossing over is exchange of genetic material between two homologous chromosomes. It is an enzyme-mediated process. Enzymes involved is called recombinase. Crossing over leads to recombination of genetic material which is completed by the end of packetine, leaving chromosomes linked at the site of crossing over. Diplotein. Beginning of diplotein is recognized by the dissolution of synaptomal complex and tendency of the recombined homologous chromosomes of the bivalent to separate from each other except at the site of crossovers. This X-shaped structure's site of crossing over is called chiasmata. In oocytes of some vertebrates, diplotein can last for months or years. Dictyotein. Diakinesis. This is marked by terminalization of chiasmata. Chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears and nuclear envelopes also breaks down. Diakinesis represents transition to metaphase. Metaphase 1. Bivalent chromosomes align on the equatorial plate, double metaphasic plate. Microtubules from the opposite poles of the spindle attach the kinetochores of homologous chromosomes. Anaphase 1. Homologous chromosomes separate while sister chromatids remain associated at their centromere. Telophase 1. The nuclear membrane and nucleolus reappear. Cytokinesis follows and this is called dyad of cells. Interkinesis. It is the stage between two meiotic divisions. It is generally short-lived, no DNA replication. It is followed by prophase 2. Meiosis 2. Equational phase. Resembles a normal mitosis. Prophase 2. It is initiated immediately after cytokinesis. The nuclear membrane disappears by the end of prophase 2. Chromosomes become compact. Metaphase 2. Chromosomes align at the equator. Microtubules from opposite poles of the spindle get attached to the kinetochore of sister chromatids. Anaphase 2. Splitting of centromere allowing chromosomes to move towards opposite poles of cells by shortening of microtubules attached to the kinetochores. Telophase 2. Meiosis ends with telophase 2. Two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrads of cells. Significance of meiosis. Conservation of specific chromosome number of each species is achieved across generation in sexually reproducing organisms. Paradoxically, results in the reduction of chromosome number by half. Increases genetic variability in the population of organisms, which is important for the process of evolution.